All right, great. Well, thank you everyone for coming out tonight. My name is Chris Monk. I'm a faculty member of the Department of Psychology. So tonight, I'm very pleased to introduce Dr. Natalie Tronson, and she'll be talking to us about some of the recent research she's been doing related to COVID, as you see. So Dr. Tronson is an associate professor of the Department of Psychology here at the University of Michigan. She completed her undergraduate degree at the University of South Wales in Sydney, Australia in psychology. And then she came here to the United States and received her PhD in behavioral science at Yale University. And then following that, she was a postdoctoral fellow in the medical school at Northwestern University. And then after that, she came to us and joined us at the University of Michigan in 2012. So Dr. Tronson's research examines the molecular mechanisms of learning and memory, the factors that lead to changes in memory, and the role of memory problems in psychiatric and neurological disorders, including post-traumatic stress disorder, or PTSD, and post-operative cognitive decline. So Dr. Tronson has received funding from the National Institutes of Health to study important topics such as this with COVID, and how COVID is related to inflammation as well as dementia. She also has received a Public Engagement Faculty Fellowship from the U of M, the Oscar Stern Award from the U of M Depression Center, and she received a very competitive American College of Neuropsychopharmacology Travel Award as well. Dr. Tronson's talk is titled COVID-19, Inflammation and Risk for Long-Term Memory Decline. Please join me in welcoming Dr. Tronson.
Thank you, Chris, for that very kind introduction and for organizing this series this year and last. And thank you to the Ann Arbor District Library for hosting us, and Jackie and Tyler for organizing the event and doing the AV as well. So I'm talking about COVID-19, inflammation, and risks for long-term memory decline. And before I start, I want to give some special thanks to the people who led this work in my lab. This is Amy Choi presenting this work at a conference in the fall, Pipin Kalind, who's my lab manager, and Daria Chesalova, who did an earlier version or some of the basic work on this and is now a research scientist in pharma industry. So when we're talking about COVID, or we're talking about the SARS-CoV-2 virus, one of the things I just want to make a note of up front is that this rarely infects the brain. It's not unique to viruses on its effects on the brain, even without that infection. I also want to say that COVID on its own does not cause dementia or cognitive decline, although it does cause cognitive symptoms like long COVID. And that includes brain fog, memory problems. But what might be true, and what is likely to be true, we don't know yet, is that COVID likely increases risks of dementia that already exist, dementia or cognitive decline that already exist. And that might include a genetic predisposition, lifestyle factors, and all of the other things that we know that contribute to dementia and Alzheimer's disease. And so what I'm interested in doing work on at the moment is what are the long-lasting effects of COVID on the brain? What is happening after recovery from illness that might persist in the brain and trigger dementia and cognitive decline later on? And I was joking before that there's always a half slide in here. This is it. But what I'm going to be talking about is what is inflammation anyway? What do we mean by that? And what does it have to do with the brain? How can we study this question in mice? And then we're going to talk about implications and future questions. So, inflammation, and you hear this a lot, and often you hear that inflammation is bad and it's something that we want to get rid of. I'm going to tell you that inflammation is absolutely essential. Without inflammation, you die. It's a fast response to tissue damage or invasion by bacteria and viruses. And this is in the periphery somewhere, in some piece of skin. The inflammation itself is what you think about when you have a cut or a bump. It's heat and swelling, redness, pain, itchiness maybe. As the immune cells flood in to help heal damage and clean up the mess and destroy any viruses and bacteria. And when I'm talking to my 5-year-old about this, and we've had a lot of talks about the immune system over the last 3 years, I tell her that the immune cells and the immune system are kind of like ninjas. And I'm not the first person to describe it this. There's this great anime series called Cells at Work who have these pictures. And those are the cells that are coming in to do this work. But who tells them what to do? Where are they getting their information from? How do they know where to go and what to do and what particular response is required, be it for injury or for a virus or bacterial infection? So it turns out, importantly, in the body, we have very specialized receptors in the tissue that recognize when damage has occurred, mostly because cells are broken open, and so things that should be inside the cells and no longer inside the cells. And they recognize damage or bacteria or viruses, and they're specific to each one. And we call these DAMPs or PAMPs, which are ridiculous names for receptors, but that's damage-associated or pathogen-associated molecular patterns. Some of these we call toll-like receptors, which is a term that will come back. These activate inflammation via a pattern of cytokines. So cytokines are molecules, they're signaling molecules. Those are the ones that signal which immune cells to show up, what kinds of patterns should happen, what needs to happen there to heal or to fix the damage or the inflammation or the invader that's there. So that's great. But what does that have to do with the brain? So how do you feel when you're sick? This is an audience participation slide. So how do you feel when you're sick? Headache. Okay, great. Tired. Nauseous. Heartburn. And there's many others. Fever, cranky, unhappy, lethargic, brain foggy, can't imagine feeling good again, unfocused. All of these things happen in your brain, including the fever that's controlled by your brain. And this is important because cytokines in the brain make you feel sick, or at some level they do. So inflammation in the body, when we have an infection or even major injury, they cause cytokine activation in the brain that basically makes you feel bad enough that you have to stay home in bed and away from people. And that lets you rest, allows the immune system function properly, and keeps you away from other people so you don't spread whatever it is you have. So cytokines are how your immune systems communicate with your brain, and that's really, really important. But the immune system in the brain does more than just sickness. Cytokines and other cells, including microglia and astrocytes, are really essential for basic neural functions. And they do this by wrapping around and interacting with neurons, and in particular, we're going to be talking about this part of the neuron where they connect together called the synapse. So this is where the neurons connect together. It's how we form memories. It's how they talk to each other and make muscles move and make us think and all of those good things. So in the brain, microglia hang out and they surveil the environment to make sure everything's fine. They also clean up injured tissue. Astrocytes wrap around the synapse and clear excess neurotransmitters so you don't end up having seizures. They provide energy for neurons, and these things are happening constantly. They're making sure the brain is working properly. Microglia also remove damaged synapses. And astrocytes and microglia regulate communication between neurons. So these can be both at normal times when there's no sickness, but they also happen during sickness. And so they're well placed to sort of mediate this idea of how does being sick, how does inflammation, how does the immune system change how your brain is functioning, as particularly with this complex cognitive functions like memory. But one of the things we know is that inflammation during sickness is temporary. You get sick, you have inflammation, you recover, it goes away again, ideally. And despite that, many illnesses, including COVID, cause lingering syndromes, including brain fog and memory impairments. And we know a lot about this from post-heart attack, after surgery, major surgery will also trigger these in many patients. We also know, and this is work done by somebody else in the psychology department, that cognitive decline during aging is associated with not just inflammation during aging itself, but inflammation earlier during adulthood. So if you're looking at patterns of inflammation in the blood of a population of people and you're measuring them during midlife and then again a decade or two later, that inflammation during that midlife period before they have any memory symptoms is associated with memory decline later on. And that's important because we also know that inflammation is both a consequence of having Alzheimer's disease and other kinds of neurodegenerative disorders, but it can also be a trigger for Alzheimer's-like pathology, especially in the animal models that we use. So long COVID and post-viral syndromes, but long COVID has taken really a lot of our intellectual energy recently. And that's because more than half of patients that recover from COVID report lingering fatigue, brain fog, and other symptoms, at least in the short term. After 6 months, that drops down to about 1 in 20. That's still an enormous number of people. I also want to note here that this is not sex unbiased or gender unbiased. More than 60% of long COVID patients are women, and this is actually consistent with a lot of the other dementias that we know, particularly Alzheimer's disease. And we also know that risk increases with repeated infection. Now, I don't want this to be all doom and gloom here. I do want to add this that I just read and I was very excited about, that there does seem to be a decreasing risk associated with more recent waves of COVID, and it's not clear whether that's due to the virus itself changing and the patterns of inflammation changing, or more likely, much higher levels of immunity. But COVID here is special, not because it does something different from other viruses, but because of the enormous number of people that have been exposed and have had the disease. And that's over 673 million people, according to Google, worldwide, as of today, and more than 102 million people in the US itself. So it's special because of the size of the pandemic, not because it has a lingering syndrome. That seems to be fairly common across a lot of different illnesses. So in my lab, our big question, big, not going to answer it this year kind of question, is how does COVID or other illnesses trigger long-lasting impairments in memory and cognitive function? And our working idea is that a virus, here, COVID, is activating cytokines and inflammatory signaling, and that this is causing changes in the brain that cause these long-term increased risks for cognitive decline and dementia. Now, what we want to know is what those processes are. So we can expand that a little bit to also include what happens in the decades to come. We don't know. We've just had this 3 years worth of pandemic. Are we going to see an increase in Alzheimer's disease and other dementia risks later on? And with so many people affected by COVID, how is this going to change what is already predicted to be a surge in Alzheimer's disease? So a specific research question is, what is the role of inflammation itself? We're in this project ignoring the viral infection, which has its own special consequences, but we're just thinking about these inflammatory consequences that's triggered by viruses like the SARS-CoV virus. And what does that do to the brain? So we can think about this. We know that this virus attaches itself to this specific viral receptor, Toll-like receptor 7, and it has a specific antiviral inflammatory response of cytokines and other cells, and we think that that's going to lead to memory impairments, and that's our major focus for the work that we do. So how do we study this? And how do we study this in the animals that we use in our lab, which is mice? So we can inject a number of different kinds of substances, each of which can trigger this inflammation. None of these are viruses, none of them are bacteria, and we can do things that trigger different kinds of receptors to mimic the inflammation by a specific type of virus or bacteria. So we've used this one. This is probably the most commonly used in inflammatory research, lipopolysaccharides. We can use that mimics bacteria infection. It's made from E. coli. We can use this one that mimics the inflammation that's triggered by something like the flu, a double-stranded virus. But in this project, we're going to talk about R848, which is an antiviral medication, which is why it's available. It activates this receptor, the same one that's activated by the SARS-CoV virus at first, toll-like receptor 7, and triggers, we think, a similar pattern of cytokines. It turns out, though, lots of people have done this with a variety of different inflammatory injections. And so we know a lot about what happens in the brain during an illness or during inflammation. But illnesses aren't a one-shot thing. It's not the case that you have inflammation, a sudden spike in inflammation that lasts 24 to 72 hours and then it's gone. They often last for weeks. And there's a slower onset and a waning or up and down sort of inflammation pattern. And we don't know what that looks like in the brain either. The immune system and the brain also both change with experience. And this is really important here because any kind of experience, whether that's stress, whether that's learning something new, whether it's coming to this talk, or whether it's the experience of an immune challenge, you're going to have plasticity in the brain and for the immune response, for immune challenges also in the immune system. So what we want to know is we want to know what happens next. What happens after the inflammation is gone and the changes in the brain and in the immune system persist. And we want to know what those are because we really don't know what they are yet either. So we do this by sort of mimicking 2 weeks worth of sickness. We take our animals and they get an injection of this inflammatory trigger, that's the R848, every 3rd day over a 2-week period. They don't get very sick here, none of them die, they just they lose a little bit of weight after each of the injections, so we know it's having something of an effect, but they recover, and then at the end, they recover and gain weight again. And then we just wait. We just leave them in their home cages, we change their cage, we do our regular animal care, they have food and water ad lib, they just hang out and wait. And I'm going to be showing you data from this 2-month period because we're really interested in these long-lasting effects. We do a variety of behavioral testing here. And in animals, we can test memory, we can test changes in emotion-related behavior like anxiety. We can look at things that mimic depression-like symptoms in animals. And we can look at see what's changed in the brain and in the peripheral immune system as well. And I'm today going to be talking about this data from the antiviral that mimics the COVID inflammation, and I'm going to be talking about some of the behavioral data and particularly the memory tests that we've been doing. So our 2-week immune challenge data causes memory deficits that persist for months. So all of this data is from that 8-week time point, so they've been sitting around for 2 months afterwards, and we've used 2 tests. The first is novel object recognition. So here we take the mouse and we put them in a box that has 2 objects that are the same. Now mice are really curious creatures. I don't know if you've ever seen one running around their house. They like to explore, they like to climb on things, they like to sniff at new objects. So they'll do that, and then we take them out of the box and we put them back a little while later, but with one of those objects is switched out for something new. So now they have one object that they've seen before and one novel object. And because they're curious creatures, if they remember the original object, they'll go and spend more time with the new thing. And that's pretty reliable. What I'm showing you here is in both female and male animals, there's white bars, there's gray bars actually, are our control animals. They're both showing a preference for exploring the novel object. They're remembering the original one and they're exploring that just fine. The animals that had this previous treatment 2 months ago with this inflammatory challenge are not showing that preference at all. So it seems like their memory for this object has been decreased. They're showing memory impairments. We do another form of memory testing. We do a few, but one of them, the one that I'm going to show you today is called context fear conditioning. Now, fear conditioning is always a really strong form of memory, and that's important because learning to avoid things, learning things that are bad, learning about danger is really important for survival. And so for this, we put animals into a small box about this big that has wood floors, and they explore the box for 3 minutes, and then they get a mild shock to their foot off from the grid. They don't like it. It doesn't cause any lasting damage or major pain. It's 2-second foot shock. That's it. We put them back in their home cage. And this is when we put them back. And what we can measure is a whole variety of defensive behaviors or avoidance kind of behaviors. So if you've ever heard the term scared stiff, That's what we're measuring in mice. We call it freezing. They stay very still. So instead of exploring like we'd normally expect in a box, they just stay crouched like this. And we can measure this as just the percentage of the amount of time they spend doing that when we put them back in the box. And I have two different age groups here. These ones are animals that had the immune challenges, young adults, and these ones were middle-aged animals, about 9 months old. And what we can see here is that the young female animals show a deficit, a decrease in the amount of fear memory they are showing here. In the males, for some reason, our middle-aged, our older males are showing more fear in general, but only the males are showing this decrease, and the females look intact. And there's some interesting reasons why we might be seeing differences at different age points here, mostly because estradiol signaling in females interacts with immune function. But this is really interesting because we know there are really important effects of both age and sex in vulnerability to memory decline and memory impairments over a life course. So one of the important questions here for understanding what's happening, and this is the question we always get asked by people who are primarily neuroimmunologists, is, well, okay, that's great, but they've had this inflammation challenge. It's probably just lingering inflammation. You've just got cytokines that are still there, and that's what's causing these deficits. So we tested that. So here, our white bars are animals that got an injection of saline. And these black bars are 4 hours after the injection of this inflammatory R848. So we expect and we see this increase in a lot of these different cytokines while that drug is on board. Great, our drug works, excellent. But when we measure the same thing 8 weeks later, so these are animals in the pink bars that I'm going to show that have had the R848 8 weeks ago, they show no differences compared to these white bars, these control animals, suggesting when it's not just because we have lingering inflammation that's causing these changes in the brain to last. What we think is happening is that the inflammation has fundamentally changed what's happening in the brain or in the immune system in the brain, and that is causing memory impairments. Now, I would love to tell you the answer to what has changed in the brain. I don't know yet. But what I can tell you from this data is that inflammation is a risk factor for later memory decline, and specifically that inflammation activated through this receptor, through this mechanism that we know is activated also by the SARS-CoV-2 virus. Memory deficits in this model emerge and persist long after recovery from illness and inflammation. And what I didn't show you is if we test them soon after that 2-week exposure to inflammation, they look totally normal. Their memory is intact. So this is happening over time as they recover and after recovery. And this interaction of age and sex modifying risk for memory deficits, that's also consistent with a lot of human data with sex differences and gender differences and age differences in who's vulnerable to long COVID and other post-operative and post-viral syndromes. So what we think is happening here is that we have this inflammation, but it's not just the inflammation, it's how it interacts with other things. So it interacts with age and sex and lifestyle factors, including diet and exercise, as well as genetic predisposition here. And that together, these are changing plasticity and neuroimmune function in the brain, as well as these gene expression that's required on an ongoing basis for how cells function and how neurons function. And we have some data from a different model using a different inflammatory challenge to give us some pathways forward from previous work from our lab. And that these changes are what is causing these persistent memory impairments. And we think also probably risk for dementia as well, but we don't know. Like all good science, this research opens more questions than it answers. So what really are the consequences for COVID for age-related cognitive disease and Alzheimer's cognitive decline in Alzheimer's disease? I lured you all in here with the promise of telling you an answer for this. I don't have one yet. And as I mentioned before, Alzheimer's disease is already going to be likely a crisis with the aging population. The question that I want to think about in this work is what can we do now to prevent pandemic-related increases in risk for dementia? And how can we do that in the decades to come? Not just now, not just in 20 years' time, but over that period. All right. And with that, I have some acknowledgements. Mostly the people that I've talked about before, Amy and Pippin and Daria, who did most of this data collection, as well as all of the other people from my lab, and our funding, including a lot of this was funded by the Michigan Alzheimer's Research Disease Center, and Hank Paulson over there is sitting in the corner at this talk, so thank you specifically. And with that, I'm very happy to take questions. Yes.
Yes, you had mentioned it's not really the nature of the COVID virus, but the fact that so many people were infected by it.
Mm-hmm.
But there are other viral infections like common cold or influenza that affect lots of people. Do you see similar effects seen as a result of these viral infections?
That's a good question. The common cold, probably not, and influenza less so, although there's also a lot of population immunity to both of those things. There are some reports that suggest after the 1918 flu pandemic, there was an increase in Parkinson's-like symptoms for people who'd recovered. There are other reports, but less clear about other kinds of viral syndromes. But one of the things that we do know is chronic fatigue syndrome, or I'm going to get the name wrong, MECFS. That is also thought to be some kind of post-viral syndrome. What the specific virus is, we don't know. And the other one is Epstein-Barr syndrome, has a lot of long-term viral syndromes that have been or might be or are associated with it as well. And CFS after Epstein-Barr is pretty common as well. So there's a number of them, yeah.
So for the novel object and the freezing test, did you do it before with each animal as well as after?
So we didn't do before and after. So there's a couple of reasons, but we compared them all to animals that had the same experience without the inflammatory challenge, so the vehicle control. So fear conditioning will last for years. So we can only do that once. Novel object recognition we can do multiple times. And one of the things that we know protects against memory impairments is environmental enrichment. Right. And so environmental enrichment is basically doing things in your environment, and that could be doing crossword puzzles, or for mice chewing on crossword puzzles, or doing memory tasks. And so right now we're trying to keep this as similar as possible. And the third reason we don't like to do that is because we're waiting such a long time, particularly in our midlife animals. We're going from animals that are starting or before hormone level declines, particularly in the females, to animals that are often, in the females at least, post-estrus.
Okay. So I ask this because those same tests are personality tests, right? Their boldness and their anxiety, potentially. And so without doing a before and after, how do you know that your groups are the same for personality? And in regards to enrichment, it's the same individual difference problem, that some animals will do better and respond better. And we know that the stress will impact their immune response. So you haven't controlled for those variations, right?
So we do a lot of other control tests. So we have a lot of control tests for anxiety behaviors, which you can also think about boldness. So for those of you who are less familiar with animal work, you can think about boldness as the likelihood that animals are going to go and explore something new, as opposed to hang back because it's scary out there. And these are both absolutely reasonable responses to these situations. So what we do is we we do a couple of things. One is we're running groups of animals that are large enough that we're looking at the differences in variability. So we expect to have an overlapping population. We're getting them from the same vendors at the same time, and they're randomly selected. The other thing that we do is we do a lot of other behavioral tests. So what I didn't show you here is we have a whole suite of anxiety and depression and other memory tasks that we often do. And what's surprising to us, and what I like, because it means that we can interpret this behavioral data, is at this time point, these animals aren't showing any changes in how active they are. So if we're looking at, in any of these tests, we can look at activity level as a measure for that, which is great, because if they were still sick, they would stop running around, and that would make all of these interpretations more difficult. We also don't see any differences in what we call open field behavior, which is literally a box that's about this big square, brightly lit and with high walls. So the mice can go to the edges, which they like because they're prey for a lot of other animals, and then they will go and explore the center because they're also curious about it. But we look at this ratio of how much time they spend in the open, in the middle of the area where it's bright and scary, versus how much time they spend at running around the edge as a measure of anxiety. So more time at the edge is more anxious, less bold. More time in the center would be less bold, less anxious, or more bold, less anxious. And so we don't see any differences as a group across any of those.
Thank you, Natalie. If you were now to bring this to Alzheimer's, have you crossed these? Have you taken mice that have Alzheimer's proteins?
Yeah.
And done some of the same stresses that you talked
I am
about?
so glad you asked. We haven't yet, but we are starting to breed those animals. We're just wrapping up our protocol for this, and so Alzheimer's Association has funded us to start on that work now, and we have we're planning to use. This is for people who work on these animals, the APP1, the amyloid protein, PSEN animals. So we're looking at animals that have a sort of later 6 to 9 months old. They're 6 to 9 months old when they start to show changes in behavioral signs of memory impairments. And for some context, our young animals are about 3 months old. Our midlife animals are 6 to 9 months old. So using animals with sort of this delayed emergence of cognitive decline will allow us to do these manipulations in young animals and see if we get exaggerated cognitive decline compared to control animals at earlier time points. And then we're going to look at amyloid in the brain as well. Yeah.
I just wanted to add that in addition to Epstein-Barr virus you mentioned, HIV is a great example. You know, a real success is that we've been able to keep people from dying from immunosuppression because of HIV disease. Mm-hmm. But then people who are on long-term therapy for HIV develop what's called HIV-associated neurocognitive disorder. And we don't really understand that, but we do think it involves some inflammatory signals, and so there's probably a lot to be learned in comparing HIV disease to other viral chronic conditions that affect brain function.
Yeah, and there's several of the big groups that are doing long COVID work are HIV researchers, HIV cognition researchers for that, yeah.
Thanks so much, Natalie. That's really great. I was just wondering, obviously you're the expert on animal models. I was wondering if we can talk a little bit about the humans. I'm just curious about the brain fog data and the sex difference and what the measures were for brain fog, if it's a subjective response or if there's any, like the health and retirement study that's being run out of here at Institute of Social Research, like done this, have all this wonderful longitudinal data on midlife and older age. I wonder if there's any data coming out of there, looking at longitudinal changes from pre-COVID to post. Or
I don't know about the longitudinal data, but I hope they have that. That would be really, really interesting. The brain fog data seems to be a lot of self-report, but again, a lot of this data is still fairly new, and And there's also one of the other difficulties with this data is sort of understanding disease severity and symptom severity of the COVID infection itself and how that correlates with long COVID symptoms. And there's some papers that say there's no correlation, excluding people who end up who are hospitalized in an ICU, because ICU has its own other additional range of cognitive problems that are associated with being in the ICU. There's a group at Yale that has started to do this work, but as of SFN, when they were talking about it, they weren't talking about any of the data. But I don't know. Mostly, this has still been for the brain fog self-report in what's published. I suspect that's going to change as the data comes out.
Yeah. Okay.
Hank, do you know more than that?
No, I think the brain fog issue.
So for the audience on the recording, the answer was talking about there does seem to be some brain shrinkage in people who are recovering from COVID, and how that correlates, we're not sure, but it seems like it's probably bad.
Does COVID leave markers in RNA? If that's RNA virus, does it change RNA function?
So does it change gene expression? So does it change RNA function? That's a good question. The virus itself during the infection creates its own RNA. But afterwards, what we would expect, particularly in immune cells, and we think in the brain as well, we would expect that the cell's own RNA, which is a sort of a readout of the readout of DNA that's required to keep cells functioning. We expect those to have changed a little bit, and so that might change. That's a mechanism to change long-term function of the cell. Does that answer the question?
Do we have any more questions?
Hi, Natalie.
Hi, Chris.
Great talk. You mentioned that when it comes to like the populations of animals you're studying, specifically with the females, you have to take into consideration that their hormone levels are changing. Can you talk a little bit more about that? And also in the context of humans, when women go through menopause, that is a rather big shift in their endogenous hormone levels. And do you think there could be some interaction there?
Yeah. So there are big sex and hormonal effects on immune function, as we know. There are also differences between males and females in animals, in their neuroimmune activation in the brain. Those are probably also hormone dependent, but and we also know that there are changes both in immune activation states and in the brain after menopause, or in mice after estropause. And part of that is that the estradiol is, estrogens, are often very protective and also can exaggerate some pathways. Exactly how those two things interact in the brain, we don't know, but what we do know is from Alzheimer's disease patients and from Alzheimer's disease models, is that menopause marks a pretty abrupt transition into higher rates of Alzheimer's in women and more faster decline. So what I would expect in the Alzheimer's model mice with the immune challenge is that after during aging, after estropause, we would see a marked decline and a marked exaggeration of effects, which in the females. And whether we would also see the same things in males would be interesting, and that would suggest maybe a protective effect in non-Alzheimer's genotype, but maybe an exaggerated effect in animals and possibly people who have Alzheimer's markers. There is a weird thing in the neuroimmune literature where the people who come from a neuroscience background almost exclusively study males, and the people from the immune background almost exclusively study females, and so we have a hodgepodge of data often, and we don't really know how those two things interact. So those questions are excellent ones, and we don't have answers yet, but stay tuned.
Okay, more questions? Anybody else? No? Okay, thank you so much. It was wonderful.
Thank you all for all of the questions.